This video is going to review how to do a simple monohybrid cross using a punnett square. Remember that punnett squares represent meiosis and fertilization between two parents. When we use a punnett square to solve a genetic cross, it helps us to see the patterns of inheritance that exist. There are 5 steps that help to make those patterns clear. First, we use any information that's available to make a key. This will tell us the dominant allele and the recessive allele and the trait that they represent. Next, we use the available information to find the genotypes of the parents. In this case, we have one parent that is heterozygous and one parent that is homozygous recessive. Next, we set up and solve the punnett square. Each of the parents are diploid, so their alleles will separate away from one another and wind up in separate gametes. We represent this by showing each of their alleles in separate rows. This parent, also diploid, is going to separate his alleles on separate columns. The individual squares inside the punnett square represent fertilization. So if these are the eggs represented by mom, and these are the sperm represented by dad, and this egg meets this sperm, we will get the genotypic combination in the zygote that is heterozygous. If this egg meets this sperm, we will get this combination in the zygote, which is homozygous recessive. When this egg meets this sperm, we again will get a heterozygous combination in the zygote, and when this egg meets this sperm, we will get another homozygous recessive combination. Last, we're going to interpret the data in the punnett square to find our genotypic ratios and our phenotypic ratios for our offspring. There are 3 possible genotypes for our offspring. They could be a big A combination, a big A combination, or a little a combination. If we look in the punnett square, we can see that there are no homozygous dominant combinations in our offspring. 0 out of the 4 squares are big A. If we look again, we can see that there are 2 heterozygous combinations. 2 out of the 4 squares are big A. Lastly, we can see 2 squares that are homozygous recessive. That's 2 out of 4. So these are our ratios for the genotypes of our offspring. The last thing that we need to do is to interpret the phenotype. Now, the phenotypes can actually be seen in the key. So we're going to go ahead and we're going to copy down the 2 possible phenotypes, which are yellow and white. Now, in order to have a yellow phenotype, because yellow is dominant, the genotype has to have a dominant allele. There are 2 genotypes that have dominant alleles, the homozygous dominant and the heterozygous. We can see that the homozygous dominant doesn't have any occurrence in our punnett square. However, we have 2 out of 4 that are big A. So 2 out of the 4 possible offspring are going to be yellow. We also know that in order to be white, we're going to need to have only recessive alleles. That is going to be a homozygous recessive genotype. And we know that 2 out of the 4 offspring have the homozygous recessive genotype. This tells us that half of our offspring will be yellow and half of our offspring will be white. Let's try a problem. In humans, dimples are dominant over freckles. Cross a homozygous dominant person with a heterozygous person. This is a very simple monohybrid cross. So we're going to start off by making our key. In our key, we can choose whatever letter we like. I'm going to use D. Big D is our dominant allele, and it tells me in the problem that dimples are dominant. So big D is for dimples. That means little d is going to be for no dimples. The second thing I'm going to do is I'm going to write the genotypes of my parents. So there are 2 parents here, a homozygous dominant person and a heterozygous person. So my parents in this case are going to be homozygous dominant. That's going to be big D. And the second parent is going to be heterozygous. That's going to be big D, little d. The next thing that I'm going to do is I'm going to set up and solve my punnett square. Remember that my alleles separate away from each other. So here is big D, each one on its own row. And here is big D, little d, each one on their own column. So I'm going to take these and I'm going to fill out and solve my punnett square. Okay, so I now have my punnett square in place. Now I'm going to figure out my genotypic and phenotypic ratios, and I'm abbreviating both of those. I can see that I have two different genotypes. I have a big D genotype in the punnett square, and I have a big D, little d in the punnett square. 2 out of the 4 of the genotypes in my punnett square are big D, and 2 out of the 4 are big D, little d. Now you'll notice that I'm not writing little d because there are no occurrences of that. It's not necessary to put it in there, but if you would like to put it in there, you can. My phenotypes, I have 2 phenotypes. I can have dimples, or I can have no dimples, and I got that from looking at my key. And now I'm going to look at my genotypes. Remember that anything that has a dominant allele is going to have the dominant phenotype. So this has 2 dominant alleles, and this has a dominant allele, which means I'm going to add these 2 together. 4 out of my 4 are going to be dimples. That means 0 out of the 4 will have no dimples. This problem is a little bit more complicated. The gene for telescopic or pop eyes is recessive in goldfish. If a homozygous dominant fish is mated with a pop-eyed fish, what are the probable genotypes and phenotypes of their offspring? Now, regardless of how complex the problem actually seems, we're always going to use the 5 steps. So I'm going to start out again with my key. It tells me in the problem that pop eyes are recessive. So I'm going to use the letter E for my traits, and I know that pop eyes is recessive. That means no pop eyes is dominant. My parents are shown to me in the problem. It tells me that I'm going to have a homozygous dominant fish mated with a pop-eyed fish. So I'm going to start with my homozygous dominant. That's going to be big E. And I have a pop-eyed fish. Now, they're not telling me heterozygous or homozygous, but what they are telling me is that pop eyes are visible, and that is a recessive trait, which means that this has to be a homozygous recessive organism. Next thing I'm going to do, third step, I'm going to set up and solve my punnett square. So I've got big E on the side. I have little e on the top. I'm going to solve the square. So now my square is done, and now I'm going to interpret it. So I'm going to start with my genotypes. I only have one genotype represented here, which is a big E, little e, and 4 out of 4 of my offspring are big E, little e. That means every single one of them is going to have that genotype. My phenotype, I have 2 possibilities. I have no pop eyes, and I have pop eyes. And if I look up here at my genotype, remember, if there is a dominant allele, it's going to show the dominant trait. So my dominant allele is in all of my offspring. That means that there are going to be 4 out of 4 no pop eyes, 0 out of 4 pop eyes. This problem is a little bit more complex, but it also asks a very specific question. The ability to smell asparagus in your urine is recessive. If the mother is homozygous dominant for this trait and the father is homozygous recessive, how many of the children will be able to smell asparagus in their urine? So again, I have 5 parts to the problem. I'm going to start with the key. It tells me that the ability to smell asparagus in your urine is recessive. So I'm going to choose big A and little a, and smelling and I'm just going to abbreviate the trait as a smelling. Smelling is recessive and dominant is not smelling. I'm going to then figure out the genotype of the parents. They tell me that the mother is homozygous dominant. That's going to be big A, big A. And they tell me that the father is homozygous recessive. That's going to be little a, little a. So now I'm going to set up and solve my Punnett square. So I've got big A and big A on the side and little a on the top. And I'm going to solve the square. Now I'm going to interpret the square. My genotype is only one type. That's big A, little a. And I have 4 out of 4 of those. My phenotype, I have 2 different kinds. I have no smelling, not smelling, and I have smelling. Remember that if it has one dominant allele, it's going to show the dominant phenotype. And since these are heterozygous, this is going to be 4 out of 4 with the dominant phenotype, which is not smelling, and 0 out of 4 with the recessive phenotype, which is smelling. Now that we've done a number of practice problems, you should feel pretty confident in attacking the problems that are in the practice. Remember that when you finish, the key is also on Schoology, so you can check your answers and make sure that you are doing it correctly. And if you're having any difficulty setting up the problem or solving the problem or interpreting the genotypes and phenotypes, take the time to consult with your teacher and do some additional practice problems.